For more than 3 billion years, life on Earth existed in forms so profoundly simple that if you could somehow travel back to witness those ancient oceans, you would struggle to recognize anything as living at all. The vast expanses of water that covered most of our planet were filled with single-celled organisms, bacteria and archaea, drifting endlessly in the currents like invisible specks of dust suspended in an enormous blue void. These microscopic beings were the sole inhabitants of a world that knew nothing of complexity, nothing of the intricate dance between predator and prey, nothing of the sophisticated sensory systems and behavioral patterns that would one day characterize animal life. The seafloor during this immense stretch of prehistory was carpeted with microbial mats, thick biological layers composed primarily of cyanobacteria that stretched across the shallow coastal regions like endless living quilts in shades of green and brown. These mats absorbed sunlight with patient efficiency, releasing oxygen through photosynthesis in a slow, methodical rhythm that had continued uninterrupted for eons beyond human comprehension. There were no animals moving through these waters, no predators stalking through the depths, no eyes watching for danger or opportunity. Life was abundant, thriving even, but it was monotonous in a way that defies imagination. The planet functioned as a vast garden tended exclusively by microscopic gardeners who possessed no awareness of the complexity that would eventually emerge from their descendants through the relentless mechanisms of evolution and natural selection. This was the world as it had existed for the overwhelming majority of Earth's history, a world where evolutionary change operated at a glacial pace so slow that 1 million years might pass with barely any noticeable difference in the forms of life that inhabited the planet. Life was locked in a kind of biological stasis, as though it had reached a comfortable equilibrium and found no compelling reason to push beyond the boundaries of simplicity. If you could have somehow visited those ancient shores, walking along beaches composed of sediments that have long since been ground to dust, compressed into rock, uplifted into mountains, and eroded away again in cycles repeated countless times, you might have reasonably assumed that this simple existence was all that life would ever be, all that it was capable of becoming. And then, approximately 541 million years ago, something changed. Within a span of roughly 20 million years, a duration that seems impossibly long when measured against a human lifetime, but registers as barely a fleeting moment in the vast expanse of geological time, the oceans underwent a transformation so dramatic and so comprehensive that scientists still struggle to fully explain it even after more than a century of intensive study. The fossil record, which had been sparse and dominated by simple, soft-bodied organisms that left barely any trace of their existence, suddenly exploded with an astonishing diversity of complex life forms. Creatures with hard shells appeared for the first time, their bodies armored in calcium carbonate that could be preserved in the rock record for hundreds of millions of years. Trilobites, those distinctive arthropods with segmented bodies and compound eyes containing hundreds of individual lenses, began crawling across the seafloor in vast numbers, leaving tracks and trails that would fossilize and provide evidence of their behaviors. Predators emerged from the evolutionary crucible, some reaching lengths of 1 meter or more, hunting with sophisticated vision systems and grasping appendages that could tear apart prey with terrifying efficiency. The first chordates appeared in the water column, small swimming animals with a flexible rod running down their backs called a notochord, and from this humble beginning would eventually arise every vertebrate animal on Earth, from fish to amphibians to reptiles to birds to mammals, and ultimately to human beings capable of contemplating their own origins. This event, which scientists now call the Cambrian Explosion, was not gradual in the way that Charles Darwin had imagined when he first formulated his groundbreaking theory of evolution by natural selection. It was not the slow, steady progression of small changes accumulating imperceptibly over vast stretches of time that Darwin's theory seemed to predict. Instead, it was an eruption of biological complexity, a kind of evolutionary Big Bang that produced virtually every major animal body plan that we see in the modern world. And it happened so quickly, at least by the standards of deep time, that it seemed to violate the very principles of evolutionary theory that Darwin had so carefully articulated. Darwin himself was acutely aware of this problem, and he considered the sudden appearance of complex life in the Cambrian fossil record to be one of the gravest objections to his entire body of work. If evolution proceeded through countless small modifications accumulating gradually over vast stretches of time, as his theory suggested, then why did the fossil record show such an abrupt and dramatic appearance of complex life? Where were the transitional forms, the intermediate steps that should have been preserved in the rocks if evolution had worked the way Darwin believed it must? These questions troubled Darwin deeply, and he devoted considerable space in On the Origin of Species to addressing what he called the imperfection of the geological record, arguing that the transitional forms must have existed but simply had not been preserved or had not yet been discovered. For over a century following Darwin's death, paleontologists searched diligently for answers to these vexing questions. They excavated fossil sites in some of the most remote and inhospitable corners of the world, places where ancient seabeds had been lifted high into mountains through the inexorable forces of plate tectonics and then exposed by millions of years of erosion. They developed increasingly sophisticated techniques for analyzing the chemistry of ancient rocks, measuring oxygen levels and carbon isotopes to reconstruct the environmental conditions of the Cambrian seas with ever greater precision. They studied the genetics of modern animals in minute detail, looking for molecular clues about when key innovations first appeared in the evolutionary history of life. And slowly, painstakingly, through the accumulated efforts of thousands of researchers over many decades, a coherent picture began to emerge from the fragmentary evidence. The Cambrian Explosion, it turned out, was not a random accident, not merely a stroke of cosmic luck or an unexplainable mystery. It was the result of a perfect storm of conditions, a remarkable convergence of environmental changes, evolutionary innovations, and ecological pressures that had been building steadily for hundreds of millions of years before the explosion itself occurred. The planet had been preparing for this transformative moment, assembling all the necessary pieces like a complex puzzle, and when everything finally aligned in just the right configuration, life seized the opportunity with a burst of creativity and adaptive radiation at a speed that would never be matched again in the subsequent history of our planet. The evidence for what happened during the Cambrian explosion comes from many different sources, each providing a unique window into this ancient world. Scattered fossils found in rocks around the globe offer glimpses of the creatures that lived and died during this period. Chemical signatures preserved in ancient sediments tell us about oxygen levels, ocean chemistry, and climate conditions. The genetic relationships between modern animals, painstakingly reconstructed through comparative DNA analysis, allow us to trace lineages backward through time and estimate when different groups diverged from their common ancestors. But if you truly want to see the Cambrian world, if you want to stand face to face with the actual creatures that emerged during this explosion and understand the ecosystems they inhabited, there is one place above all others that provides an unparalleled view into deep time. High in the Canadian Rockies, in British Columbia's Yoho National Park, there exists a thin layer of rock known as the Burgess Shale that has preserved a moment in the Cambrian period with such extraordinary clarity that it fundamentally changed our understanding of early animal life when it was first discovered and studied. The story of the Burgess Shale begins on a late summer day in August of 1909, when an American paleontologist named Charles Doolittle Walcott was riding on horseback through the mountains near the small town of Field, British Columbia. Walcott was already a distinguished scientist, serving as the secretary of the Smithsonian Institution, and he had spent many years of his career studying Cambrian fossils from various sites scattered across North America. On this particular afternoon, as Walcott and his wife were making their way down a mountain trail, one of their horses allegedly dislodged a block of shale that split open to reveal the fossilized impression of a soft-bodied creature preserved in remarkable detail. The exact circumstances of this discovery have been somewhat embellished over the decades, and the story of the horse's hoof may be somewhat apocryphal. But what truly matters is that Walcott immediately recognized that he had stumbled upon something extraordinary, something that would prove to be one of the most important paleontological discoveries ever made. Over the following summers from 1910 through 1924, Walcott returned to the site again and again, excavating a quarry high on the slopes of what is now called Fossil Ridge. He worked under conditions that would test anyone's physical endurance and determination at an elevation of over 2,300 meters, where the weather could turn violent without warning, and where the working season lasted only a few short months each year before winter made the site completely inaccessible. Despite these formidable challenges, Walcott and his team managed to extract more than 65,000 specimens from the Burgess Shale, carefully wrapping each precious fossil and shipping them back to Washington, D.C., where they were meticulously cataloged and stored in the collections of the Smithsonian Institution. What Walcott had discovered was a fossil site of truly exceptional preservation, a place where not just the hard shells and skeletons of ancient animals had been preserved, but their soft tissues as well, the muscles and gills and digestive tracts that normally decay within days or weeks of death and leave absolutely no trace in the geological record. The Burgess Shale formed roughly 508 million years ago during the middle of the Cambrian Period, and the conditions that allowed it to preserve soft tissues were both unusual and remarkably precise. The animals whose fossils we find in the Burgess Shale lived in a shallow marine environment near the base of a massive underwater cliff, part of the ancient continental margin where the seafloor dropped away steeply into much deeper water. Periodically, perhaps triggered by storms or earthquakes, or simply by the instability of sediments accumulated on steep slopes, underwater mudslides would cascade down this cliff face, picking up animals from the shallows and carrying them rapidly into the deep oxygen-poor waters far below. In this low oxygen environment, the bacteria that would normally decompose organic tissue could not survive or reproduce effectively, and the fine mud that buried the animals protected them from scavengers and from the mechanical disruption that waves and currents would have caused in shallower water. Over millions of years, as more and more sediment accumulated on top of these buried organisms, the tremendous pressure transformed the soft mud into hard shale, and the soft tissues of the animals were gradually replaced, molecule by molecule, with minerals, creating fossils that preserve details down to the level of individual cells in some remarkable cases. When you examine a Burgess Shale fossil in a museum display case or in detailed photographs, what strikes you immediately and powerfully is the extraordinary level of anatomical detail that has been preserved. You can see the individual segments of arthropod bodies, the delicate feathery gills that would have extracted dissolved oxygen from the water, the compound eyes with their hundreds of individual facets arranged in precise geometric patterns, and even, in some truly exceptional specimens, the remains of the last meal still visible in the digestive tract of a predator. These are not vague impressions or fragmentary remains that require enormous amounts of interpretation and imagination to understand. These are complete animals frozen in time, preserved with such fidelity that we can reconstruct not just what they look like externally, but how they lived, what they ate, how they moved through the water, and how they interacted with other organisms in their environment. The Burgess Shale opened an unprecedented window into the Cambrian seas that revealed a world far stranger and far more diverse than anyone had imagined possible before these fossils were discovered and properly studied. Among the tens of thousands of specimens that Walcott and subsequent researchers collected from the site, certain creatures stood out for their bizarre anatomy and for the profound questions they raised about the nature of early animal evolution and the diversity of body plans that emerged during the Cambrian Explosion. The apex predator of these ancient seas was Anomalocaris, whose name means strange shrimp. Though it took many decades and the discovery of many more complete specimens for scientists to finally realize that what had initially been described as several completely separate fossils were actually all parts of a single remarkable animal. Reaching lengths of up to a meter, which was absolutely enormous by the standards of the early Cambrian world, Anomalocaris hunted with a level of sophistication that genuinely surprised researchers when they finally understood its complete anatomy and probable behavior. It possessed a circular mouth surrounded by plates that could close together like the iris of a camera, grasping appendages at the front of its body that could catch and manipulate prey with considerable dexterity, and large compound eyes mounted on stalks that gave it the ability to track movement in the water with remarkable precision and from multiple angles simultaneously. This was clearly not a primitive experiment in predation, not some crude first attempt at hunting behavior, but rather a highly refined and effective predator, an animal that required complex sensory systems, sophisticated neural processing, and carefully coordinated movement, demonstrating conclusively that the evolutionary arms race of the Cambrian had already produced highly specialized and devastatingly efficient killing machines. Equally bizarre in its own way was Opabenia, a creature so anatomically strange that when it was first presented at a scientific conference in 1972, after being properly reconstructed from Burgess Shale specimens, the audience reportedly burst into spontaneous laughter, simply unable to believe that such an animal could have actually existed in the real world rather than in some fever dream or work of surrealist art. With 5 eyes arranged across the top of its head in a configuration found in absolutely no other known animal, and a long, flexible proboscis extending forward from the front of its body and tipped with grasping claws that may have been used to capture small prey or to manipulate food items. Opabenia looked like something that belonged in a science fiction film rather than in the actual history of life on Earth. Yet the fossils were perfectly clear and remarkably detailed, leaving absolutely no doubt whatsoever that this was a real animal that had actually lived and died and left its remains preserved in the Cambrian seas. Hallucigenia earned its evocative name from its dreamlike, almost hallucinatory appearance, with prominent spines projecting from its back and what appeared to be tentacles or legs extending from its underside, though paleontologists initially reconstructed it completely upside down, a mistake that persisted in scientific literature and museum displays for many years, until better specimens were discovered and carefully analyzed, finally revealing the true orientation of this peculiar creature. The diversity of body plans preserved in the Burgess Shale extended far beyond these celebrated oddities that captured public imagination. Wiwaxia, covered in protective scales and spines, resembled something intermediate between a mollusk and an animated worm, defying easy classification and suggesting strongly that the Cambrian seas were home to experimental anatomical designs that would eventually be abandoned entirely by evolution, leaving no descendants in the modern world. Morella, sometimes called the lace crab because of its delicate appearance, became the single most abundant fossil in the entire Burgess Shale collections, a small arthropod with elaborate head shields and trailing appendages that filtered microscopic food particles from the water as it swam, demonstrating that filter feeding was already a successful ecological strategy during the Cambrian period. Perhaps most significant for our own evolutionary story was Pikaia, an unassuming creature barely a few centimeters long that swam through the water with undulating movements of its flattened ribbon-like body. Pikaia possessed a notochord, that crucial flexible rod running along its back that provided structural support for muscles and allowed for much more efficient swimming than was possible for animals without any kind of internal skeleton. This anatomical feature placed Pikaia firmly within the chordates, making it one of the earliest known members of our own lineage, a humble beginning for a group that would eventually give rise to fish, amphibians, reptiles, birds, mammals, and ultimately to human beings capable of discovering its fossilized remains and recognizing it as a distant relative. The Burgess Shale revealed a Cambrian world that was not just diverse in terms of the sheer number of different species present, but profoundly diverse in terms of the range of body plans, ecological strategies, and anatomical innovations that had evolved in a relatively short period of time. Filter feeders and active predators coexisted with burrowing organisms in free-swimming forms, while creatures that crawled along the seafloor shared the same habitats with those that drifted in the water column above. The ecological complexity of these ancient seas was genuinely comparable to that of modern marine environments, with multiple trophic levels, intricate food webs involving dozens or hundreds of species, and a remarkable variety of different adaptations for capturing food, avoiding predators, finding mates, and reproducing successfully. This was demonstrably not a primitive world of simple experimental organisms, fumbling awkwardly toward greater complexity through random trial and error. This was instead a mature, fully functional ecosystem operating according to the same basic ecological principles that govern life in our oceans today, with competition for resources, predator-prey dynamics, specialization into distinct ecological niches, and all the complexity that characterizes modern biological communities. Yet, the Burgess Shale, remarkable and revelatory as it was and continues to be, represented only the middle stages of the Cambrian Explosion, a snapshot taken when the revolution was already well underway. By the time these particular animals lived and died approximately 508 million years ago, the major innovations of the Cambrian Explosion had already been established and refined through millions of years of evolution and natural selection. To witness the explosion at its very earliest moments, to see the initial stages of this transformation when complexity was just beginning to emerge from simplicity, scientists needed to discover fossils that were even older than those of the Burgess Shale, deposits that captured life as it existed closer to the boundary between the Ediacaran period and the Cambrian period. That crucial discovery came in 1984 on the opposite side of the world from the Canadian Rockies in the Yunnan province of southwestern China, when a Chinese paleontologist named Ho Xiangguang was examining rock formations near the small town of Chengjiang and found something that would prove equally revolutionary for our understanding of the Cambrian Explosion. The rocks that Ho was studying, part of what geologists call the Maotian Shan shales, contained fossils with the same kind of exceptional soft tissue preservation as the Burgess Shale, capturing delicate anatomical details that are almost never preserved in the fossil record. But these Chinese fossils were significantly older than those of the Burgess Shale, dating to approximately 520 million years ago, and thus pushing the record of complex animal life with preserved soft tissues back by more than 10 million years, placing it much closer to the very beginning of the Cambrian period itself. What Hu had discovered was not just another remarkable fossil site to add to the paleontological catalog, but rather an unprecedented window into the earliest phases of the Cambrian explosion itself, a time when the innovations that would come to define animal life were still fresh, still experimental, still being tested by natural selection. The Chengjiang fossil beds have since yielded an extraordinary diversity of specimens, representing more than 196 different species, documented across at least 16 different phyla, representing virtually every major group of animals that existed during the early Cambrian period. The quality of preservation at Chengjiang rivals, and in some important ways, even surpasses that of the Burgess Shale, with fossils showing not only external anatomy in exquisite detail, but internal structures as well, including digestive systems, traces of nervous tissue, and even, in some truly remarkable cases, what appear to be preserved remnants of the animal's brains or central nervous system concentrations. Modern imaging techniques, particularly high-resolution micro-CT scanning that can visualize internal structures without physically damaging the precious fossils, have allowed researchers to examine these specimens in 3 dimensions with a level of detail that would have been completely impossible even just a few decades ago. The result of all this intensive study has been nothing short of a revolution in our understanding of how quickly animal complexity actually emerged during the Cambrian explosion, and just how sophisticated these early ecosystems really were, even at the very dawn of complex animal life. What makes the Chengjiang deposits particularly significant from a scientific standpoint is not merely their age or the exceptional quality of their fossil preservation, important as both those factors certainly are, but rather the fact that they contain creatures representing evolutionary experiments that were happening in real time, animals that were actively trying out different solutions to the fundamental challenges of survival in this newly competitive world where predation had become a dominant ecological force. Among the most important and scientifically valuable discoveries at Chengjiang are the early chordates, animals that possess the defining anatomical features of our own evolutionary lineage, and thus represent our own deep ancestry. Haikuichthys and Milokunmingia, both found in the Chengjiang deposits and both dating to approximately 520 million years ago, are among the very earliest known fish-like vertebrates in the entire fossil record. These were small creatures, barely a few centimeters in length, that swam through the water using blocks of muscles arranged in segments along either side of a notochord, the flexible rod that provided structural support and allowed for the characteristic undulating swimming motion. These early fish-like animals had gills for extracting dissolved oxygen from the water, sensory organs concentrated at the front of the body in what was clearly becoming a distinct head region, and a basic but clearly functional arrangement of internal organ systems that would eventually be refined over hundreds of millions of years of subsequent evolution into the sophisticated body plan shared by all vertebrate animals, including human beings. The fact that these early chordates appear in the Chengjiang deposits, so remarkably close in time to the very beginning of the Cambrian period itself, tells us something genuinely profound and rather surprising about the speed and intensity of the Cambrian explosion. These were not simple organisms by any reasonable measure. They had complex anatomy with multiple specialized organ systems, sophisticated sensory capabilities, and clearly defined behaviors adapted to their particular ecological roles. Yet they existed only approximately 20 million years after the Cambrian period began, a time span that seems almost impossibly short for evolution to have produced such a high level of complexity, starting from the relatively simple soft-bodied organisms that characterized the preceding Ediacaran period. The Chengjiang fauna demonstrates with crystalline clarity that the Cambrian explosion was not a gradual process that unfolded slowly and steadily over many tens of millions of years, as one might expect from a conventional understanding of evolutionary processes, but was instead a genuinely rapid burst of innovation and diversification that happened with truly startling speed, almost as though life had been waiting impatiently for some crucial signal, some environmental trigger that would finally allow it to explore all the morphological and ecological possibilities that had somehow remained closed off and inaccessible during the enormously long ages when complexity remained stubbornly out of reach despite billions of years of evolution. To understand why the Cambrian explosion happened when it did, and why it unfolded with such extraordinary rapidity, we need to examine the multiple factors that converged during this unique period in Earth's history. One of the most important of these factors was the steady rise in atmospheric oxygen levels that had been occurring throughout the late Proterozoic eon. For most of Earth's history, oxygen concentrations in both the atmosphere and the oceans had remained far too low to support complex animal life with active metabolisms and sophisticated behaviors. Oxygen is absolutely essential for complex organisms because aerobic metabolism, the biochemical process of extracting energy from food using oxygen, is far more efficient than any anaerobic alternative that doesn't require oxygen. A simple sponge or a small worm can survive quite adequately on relatively low oxygen levels because these organisms move slowly, have relatively low metabolic demands, and maintain simple body plans that allow oxygen to diffuse directly through their tissues without requiring specialized respiratory or circulatory systems. But an active predator like Anomalocaris, with powerful muscles for swimming at high speeds, complex compound eyes that require constant energy to maintain and operate, and a sophisticated nervous system capable of coordinating rapid and precisely controlled movements, needs abundant oxygen to fuel all of these energy-intensive physiological systems. The critical change that made the Cambrian explosion possible came during the late Proterozoic, when oxygen levels in Earth's atmosphere began to rise more rapidly than they had during previous eons, though they still had not reached modern concentrations by the time the Cambrian period actually began. Recent estimates based on multiple independent lines of geochemical evidence suggest that atmospheric oxygen may have reached approximately 15 to 18% by the early Cambrian, compared to today's level of 21%. While this difference might seem relatively small and insignificant, it actually represents a crucial threshold beyond which aerobic metabolism becomes efficient enough to support larger bodies, more active lifestyles, and the kind of complex sensory and nervous systems that characterize most modern animals. Another critical factor that enabled the Cambrian explosion was a significant change in ocean chemistry, particularly substantial increase in the concentration of calcium and other minerals that animals would soon learn to use in constructing shells and skeletons. The weathering of continental rocks during the late Proterozoic released enormous quantities of calcium into the world's oceans through the action of rivers carrying dissolved minerals from the land to the sea. This process increased the calcium concentration in seawater to roughly 3 times the levels that had prevailed during earlier periods, making biomineralization energetically feasible for the first time in Earth's history. Animals could now extract calcium from the surrounding seawater and deposit it in crystalline structures to form protective shells and supportive skeletons without expending such prohibitive amounts of energy that the strategy would be evolutionarily unviable. The appearance of the first mineralized skeletons in the latest Ediacaran period, just before the Cambrian began, marked the beginning of a profound transformation in marine ecosystems. Once some animals began building hard protective shells as defense against the newly emerging predators, an evolutionary arms race inevitably began, with prey species developing ever thicker and more elaborate armor, while predators evolved correspondingly stronger jaws, more powerful crushing appendages, and more sophisticated hunting strategies to overcome these defenses. This feedback loop, which evolutionary biologists call escalation, created intense selective pressure that drove rapid innovation in both offensive and defensive adaptations, with each new breakthrough in one domain favoring corresponding improvements in the other. The fossil record provides abundant evidence supporting this interpretation of events, showing clear signs of predation in the form of healed injuries on trilobite exoskeletons where attacks were survived, precisely drilled holes through shells made by predators with specialized boring organs, and fossilized stomach contents of predators containing recognizable fragments of armored prey species. The evolution of sophisticated vision systems fits naturally and logically into this same framework, since the ability to see would have provided enormous advantages both for predators attempting to track and capture elusive prey, and for potential prey species trying to detect approaching predators from a safe distance and escape before being caught. The genetic and developmental mechanisms that enabled this rapid diversification were also crucial to the Cambrian explosion, though these are necessarily more difficult to study directly since DNA and proteins do not fossilize under normal circumstances. Modern research in evolutionary developmental biology, a field often abbreviated as evo-devo, has revealed through comparative studies of living animals that the basic genetic toolkit for building complex bodies was already largely in place before the Cambrian explosion began, inherited from even more ancient ancestors. The Hox genes, which are master control genes that regulate the development of body segments and the positioning of appendages and organs along the body axis, are found in remarkably similar forms across an enormously wide range of animal groups, from simple worms to complex insects to vertebrates like ourselves. This striking similarity in such fundamental developmental genes across vastly different animal groups strongly suggests that these genetic control mechanisms evolved in the common ancestor of all animals, probably during the Precambrian, and were then inherited by the various evolutionary lineages that diversified so dramatically during the Cambrian explosion itself. What changed during the explosion was not the actual invention of entirely new genes from scratch, but rather the deployment of these existing genetic tools in novel combinations and in new developmental contexts, creating diverse body plans through modifications to the timing, location, and intensity of gene expression, rather than through the far slower process of evolving completely new genetic information through random mutation. This genetic flexibility meant that once environmental conditions finally favored the evolution of complex animal bodies, once oxygen levels were high enough and calcium was available and predation pressure was intense enough, the necessary developmental machinery was already available and waiting to be activated, modified, and deployed in creative new ways. Small changes in the regulation of existing genes, alterations in when and where particular genes were switched on or off during embryonic development, could produce dramatic and far-reaching changes in adult body form, allowing rapid evolutionary innovation without requiring the prohibitively slow accumulation of entirely new genetic sequences through random mutation and selection over countless generations. The Cambrian explosion was therefore not a period when the genetic instructions for building animals had to be laboriously written from scratch, starting from nothing, but rather a time when existing developmental programs and genetic instructions that had been refined over hundreds of millions of years of prior evolution, were suddenly edited, remixed, and deployed in fantastically creative new ways in response to changing environmental conditions and new ecological opportunities. The speed of the explosion becomes much more comprehensible and less mysterious when we recognize this crucial fact. Evolution during the Cambrian was not starting from zero, but was instead working with a sophisticated genetic and developmental toolkit that had already been assembled and refined over an immense span of prior evolutionary time. When we look at the totality of the evidence from multiple scientific disciplines, from paleontology and geology to geochemistry and molecular biology, a coherent picture emerges of why the Cambrian explosion happened when it did and why it unfolded with such remarkable speed. It was the convergence of multiple enabling factors, rising oxygen levels that made active, complex metabolisms energetically viable for the first time, increased calcium availability that made biomineralization possible, the disruption of ancient microbial mat ecosystems that had dominated seafloors for billions of years, creating new three-dimensional living space and new ecological opportunities, intense predation pressure that drove an evolutionary arms race, favoring continuous innovation in both weapons and defenses, and the activation and creative deployment of existing developmental genes and genetic regulatory networks that could rapidly generate morphological novelty and diversity. The legacy of the Cambrian explosion extends across the entire subsequent history of life on Earth, shaping the world we inhabit today in fundamental and pervasive ways. Every major group of complex animals alive in our modern world traces its evolutionary origins directly back to the Cambrian period, making this ancient explosion absolutely central to understanding the diversity of life we see around us. The arthropods that dominate modern ecosystems in terms of both species numbers and total biomass, including all insects, spiders, crustaceans, and their countless relatives, are direct descendants of the Cambrian arthropods that first evolved jointed appendages, compound eyes, and segmented exoskeletons more than 500 million years ago. The mollusks that we encounter everywhere from tidal pools to the deepest ocean trenches, including familiar creatures like snails and clams, as well as the remarkably intelligent and behaviorally sophisticated octopuses and squids, trace their ancestry to the early shell-bearing organisms that first evolved biomineralization during the Cambrian explosion. The echinoderms, that distinctive group characterized by five-fold radial symmetry and including sea stars, sea urchins, brittle stars, and sea cucumbers, first appeared in the late Cambrian as relatively simple organisms, but would go on to diversify extensively during subsequent geological periods. And perhaps most significantly for our own story, the chordates, represented in the Cambrian by small and seemingly insignificant swimming animals like Pikaia and Hyquithus, gave rise to the vertebrates that would eventually colonize not just the oceans, but also the land and the air, producing the extraordinary diversity of fish, amphibians, reptiles, birds, and mammals that characterizes our modern world. Rest now, knowing that you are part of an unbroken chain of evolutionary descent that stretches back more than 500 million years to a time when your distant ancestors were small swimming creatures in ancient seas, when eyes were first opening to perceive the world, when the first protective shells were forming in response to the first predators, when the fundamental architecture of animal existence was being established for all time to come. The patterns that were written into the fabric of life during those Cambrian seas are written into your own body as well, encoded in your DNA, reflected in the structure of your skeleton and organs, manifest in the very developmental processes that built you from a single fertilized cell into the complex organism you are today. The Cambrian explosion, was not just the beginning of animal diversity and complexity. It was the foundation of the living world we know and inhabit, the crucial moment when life's vast potential finally became manifest in physical form, when the basic blueprints for animal existence were drawn up, tested against the harsh realities of survival and reproduction, and refined through natural selection into forms that would prove viable across unimaginable spans of subsequent time. The story of the Cambrian teaches us that life is resilient, endlessly creative, and capable of remarkable innovation when conditions allow. The complexity emerges rapidly when environmental barriers are removed and opportunities arise, and that the foundations laid in the deep past continue to support and shape everything that comes after. Sleep peacefully, descendant of those ancient Cambrian seas, inheritor of innovations that first appeared when our planet was young and the evolutionary future stretched before life, like an open road filled with infinite possibilities waiting to be explored and realized.